The lecture exam is going to have 50 questions. And it's going to be the exact same time frame. Everything we try to make it consistent so that you get the hang of things. So 40 of the first ones are multiple choice, and they're divided evenly between these chapters: blood, heart, blood vessels, respiratory, and digestive. Okay. So 1, 2, 3, 4, 5. So you can expect about 10 questions per chapter. And then the last 10 questions are fill in the blank. And the topics are the blood, heart, blood vessels, respiratory, and digestive. And then for lab, it's going to be very similar to the other ones, 30 questions. And it's also going to be divided up between the heart, the respiratory organs, and the digestive organs. So 10 questions per those main three. And these are all identification. Okay. And in this chapter particularly, there's a lot of right and left. So make sure you identify the right and lefts. And this one's a little tricky because I noticed that there's a few more incorrect on the quizzes. When you did the right and left, don't forget it's anatomical position. Okay. So it's not how you're viewing it, but how you put your body into that picture. So with anatomical position, your right at your right hand when you're looking at it is actually the image's left. Okay, so make sure you remember that because there was a little bit more mistakes on that than that I saw than normal. So this is based on anatomical position. Okay, a lot of the structures have multiple names. Make sure you include all the parts of the names. Like for example, the inferior nasal conchae. Okay, a lot of students just input inferior conchae and didn't put nasal. So you got half a point after that on the quizzes. And that's going to be the same for the exam. So make sure you include all parts of the names. Spelling counts as always. And that's pretty much it for the lecture and lab. Okay, same as always. You want to make sure you're using the Respondus lockdown. And webcam. There were a few more issues on the exam too with the webcam. So it flags you when your face is not fully in the screen. Like if I only see the top of your head, it's going to flag you. Okay. So make sure that you are fully in the screen. Okay. And make sure that you do a thorough environmental scan. That's, what protects you. Make sure that there's nothing around you so that, you're just doing this on your own. So the environmental scan is really important. And then a reminder that you can't have anything around you, so no cell phones. There shouldn't be any paper because I can hear the paper rustling, so there shouldn't be any paper. You are allowed a blank piece of paper, but if you choose to have a blank piece of paper in your environmental scan, you have to show the front and the back of the paper that it's blank to start out with. Notes, textbook. Okay, so no resources around you. Oops. Okay. So that's the logistics for the exam. So any questions on the exams?
Not for me.
We'll go right into it then. So the first one is the cardiovascular. The cardiovascular includes the blood, the heart, and the blood vessels. So, and like you said, you should be, it's 3 chapters. It's a lot and it's old. So you want to make sure you do spend time reviewing it. We're going to start with blood first. So with blood, you want to make sure you know all the functions of blood, the components of blood, and A way that we learn about the components of blood is through hematocrit. In a hematocrit, we take your blood, we put into a tube, and we put into what's called a centrifuge. It's a little machine that spins your blood around. And when we spin the blood around, it separates your blood based on density. Okay, so the most dense goes to the bottom. Then there's a layer in the middle, and then the most abundant and also the lightest is on the top. So what are the three main components of blood based on the separation of the hematocrit? What's the most abundant and lightest? I have something in the chat. Plasma, that's right. Plasma makes up about 55% of the blood. And then the middle is what we in general call the buffy coat. And the buffy coat actually, let's do what it makes up. It makes less than 1%. And it's going to include two things. It's going to include your white blood cells. And your white blood cells are also known as leukocytes. And then it's also going to include your platelets. And it makes sense that both your white blood cells and platelets are the lowest because those are your defense mechanisms, right? So when something's wrong. So if you're healthy, nothing's going on, you should have low levels of, not low, but average levels of white blood cells. And they're lower than compared to the other components in blood, because that means that you don't have an infection, you don't have any trauma that, you need to activate the platelets or anything. So that's why they're the least abundant, okay? Then the second most abundant is going to be your red blood cells. Your red blood cells, also known as your erythrocytes, make up about 45% of blood. Okay, so those are the main components of how things are separated. So you have the plasma, most abundant, then you have the buffy coat, and then the red blood cells. Okay, and then we'll break the red blood cells down, the white blood cells down to the 5 types of white blood cells a little bit later. But for now, that's kind of the basics of the components of blood. Now, another way in which we classify the components of blood is based on what is liquid versus what's solid. So the solid components of blood would be the white blood cells, the red blood cells, and the platelets. And the liquid component of blood would be the plasma. Another name for solid, and you may see this term on the exam, is called formed elements. They're considered formed elements. They have like, form. They have shape to them, versus liquid is more fluid. Okay. So if on the test I asked you what are formed elements, you'd say red blood cells, white blood cells, and platelets. And then liquid or non-formed elements would be plasma. Okay, so those are the components of blood in general. Now, of those components, we're going to start with red blood cells. So that's going to be our kind of Roman numeral or a little letter A under 1. So red blood cells are also known as erythrocytes. Make sure you know the function, the structure, and the life cycle of a red blood cell. Okay, so let's start with functions. What are some functions of red blood cells? What are some functions of, yeah, definitely. So it transports, and you're right, Anna, it transports gases, waste, nutrients, pretty much, oh no, I'm talking about blood, sorry. Let me redo that. It transports gases. You're right. I was looking at functions of blood. Okay. All right. So let's go here. Okay. So it transports gases, specifically oxygen and carbon dioxide. Okay. So that's the main function of red blood cells. Okay, so it transports the gases. Then what is the structure of a red blood cell? What does a red blood cell look like? Yeah, we call that biconcave. So it looks like a donut because everything is about food for me. So it looks like a donut. Okay. And a side view of that, it looks like this. Because it dips in the top and the bottom, we use the term biconcave. Concave means it dips in, bi meaning there's two, the top and the bottom. So it's biconcave. Now, the reason why it's biconcave is that in the center, what do you normally find in the center of a cell? Exactly. So because it's hollow, that tells you that it has no nucleus. So a mature red blood cell has no nucleus. Okay. What's the function of the nucleus? What's the significance of a nucleus? What does a nucleus do? What does it contain? Yeah, exactly. Great job, Irene. So it contains DNA, and DNA produces protein, and protein helps to form parts because you learned about the function of proteins, right? Proteins act as catalysts, proteins do this, proteins do that. So if it doesn't have DNA, it can't produce these. And that's why a mature red blood cell, because it doesn't have because it doesn't have the DNA, it can't reproduce. Nor can it repair because it can't make the proteins, and the proteins make components of a cell, right? So it can't repair itself. And because it can't repair itself, the life cycle, it can only live for about 120 days on average. Okay. So mature red blood cells has no nucleus, so it can't reproduce, nor does it last long. It only lasts for about 120 days. Okay. What else you'll find in a red blood cell is that it has no mitochondria. What does mitochondria do? Someone remind me, what's the function of mitochondria? Yes, exactly. It produces ATP. Now, in order to produce ATP, right, in the last unit, you studied about metabolism, right? So in order to produce ATP, it needs two main things. What does it need? What do cells in your body need to produce ATP? Two main things. It needs oxygen. Yes, perfect. So it needs oxygen and glycogen, you're right. Or we can say glucose, right? Now, let's take a look at this. What's the function of a red blood cell? It's to carry the oxygen. So it wouldn't make sense. It would have an organelle that uses up what it needs to transport. Okay. And for that reason, because if it had a mitochondria, it would use up all the O2. And that's why you have no mitochondria in a red blood cell. What that tells you is that a red blood cell relies solely on anaerobic respiration. Okay, because it has no mitochondria. All right, because that defeats the point, right? If its purpose is to carry oxygen and it uses it, by the time it gets to its destination, there won't be any oxygen left. Okay. All right. Now, if we were to break down a red blood cell into its smaller elements, I'm going to move the chat over here so I can write on my screen. Okay. So if we were to take a look at a red blood cell, the functional unit of a red blood cell is what? What makes up a red blood cell? It has thousands and millions of these functional units inside of a red blood cell. Hemoglobin, exactly. So if on a test I ask you, what's the functional unit of a red blood cell, you would say hemoglobin. Perfect. Now, if we were to break hemoglobin down, it would break down into two main parts, the globin chains and the heme groups. Okay, so each red blood cell has millions of hemoglobin, and in each hemoglobin, there are 4 heme groups. Okay, these 4 heme groups have 2 main parts. It's got the heme, the pigment, and it's got iron. Okay, so the heme groups have a pigment and iron. Okay, now it's really the iron that binds to what? What does iron bind to? Just think like your car. Any metal car, it rusts because it binds to what? Not salts. Good guess though. It goes through a process called the rusting is due to the fact that it goes through a process called oxidation because metal or iron binds to oxygen. Okay, and that's how red blood cells can carry the gases. It's because the iron on the pigment, which makes the heme groups carry oxygen. Okay, where the globin chains, this is responsible for carrying carbon dioxide. Okay, so a red blood cell carries both oxygen and carbon dioxide, although mainly it carries oxygen. Okay, so if in one hemoglobin you have 4 heme groups, which means 4 iron, how many molecules of oxygen can you carry in one hemoglobin? Nope, because one hemoglobin has 4 heme groups, and each heme group has 1 iron. So there's 4 iron altogether, and which means each one can carry 1 molecule of oxygen. So altogether, one hemoglobin can transport 4 molecules of oxygen. Does that make sense? Questions? Okay. All right. So that's the structure. Okay. Now let's go through the life cycle a little bit more. So a lot of you chose to do your video on hypoxemia. So you know this already. Where are red blood cells produced? Exactly. Okay. So it's produced in the bone marrow. Okay. So you have bone marrow in the medullary cavity. You have bone marrow along the epiphysis as well. Okay. So red blood cells are produced in the bone marrow. Oh, there's two terms that you want to be able to know. Okay. So there's two terms that you want to be able to know. So one term is hemopoiesis, and the other term is erythropoiesis. Okay. So hemopoiesis means what versus erythropoiesis? So what's the difference between hemopoiesis and erythropoiesis? So hematopoiesis, uh, Poiesis, hematopoiesis versus erythropoiesis. What's the difference between those two? There is a difference. So hematopoiesis and erythropoiesis. So where hemato, just like your hematocrit, it's everything in blood, right? So hematopoiesis refers to the production of blood, and that includes red blood cells, white blood cells, and platelets, where erythropoiesis specifically refers to the production of red blood cells. Okay, so what we're talking about right now is which one, hematopoiesis or erythropoiesis? Yes, exactly. Okay, so make sure you know the distinction because sometimes students get, this is one of the more common ones that students make mistakes on because it sounds so familiar, so similar to each other. Okay, all right, so we're talking about erythropoiesis right now. So red blood cells are produced in the bone marrow, and we know what triggers red blood cell production, right? So erythropoiesis, EPO, which is that chemical that's produced by the kidney and the liver, triggers red blood cell to be produced. And when red blood cells produced, they circulate the body for how many days? 100, about 120 days, perfect. And then they're broken down. So for 120 days, they circulate around the body until finally they are broken down, and they usually get caught in the spleen. Okay, the spleen and the liver, but mostly the spleen. Okay, and that's why sometimes a nickname for the spleen is like it's the graveyard of red blood cells. That's the spleen. Okay, because the spleen has a lot of fibrous fibers that help to catch it and then break it down. So it goes to the spleen to be broken down. And when the red blood cell is broken down, it's broken down into its basic elements. So what are the functional units of a red blood cell again? Oh, EPO. EPO stands for erythropoietin. It's a chemical. H erythropoietin. If you've ever heard of like athletes doping, they inject steroids into their body increase red blood cell production, it's this steroid right here, erythropoietin, EPO. Yep. So if you break red blood cells down, what do you break it into what two main parts do you break red blood cells into? Well, actually, we'll start with what do you break red blood cells down into first, and then we'll break that down further. So red blood cells have functional units called, yep. And then these are broken down further into what? Yep. Now the globin chains, they're just protein. Okay, they're made up of protein. And if you break the protein down, what do you get? When you break the protein down to its functional units, its basic units, what are they? For this one, you have to recall the chapter on chemistry. Yes, perfect, Irene. These are the amino acids. And because they're amino acids, they're just going to be reused in the body to whatever needs protein. Okay. So whatever needs protein, they take the amino acids and they reassemble them into whatever protein it needs to be. But essentially, the globin chains in hemoglobin that make up the red blood cells are broken down into protein. The protein are broken down to amino acids, and those are just reused in the body. Okay. Then you have the heme groups. And the heme groups are broken down into two parts. What are the two parts that the heme groups are broken down into? So what do heme groups break down into? Yes, iron and pigment. Iron, we'll put iron here, and the pigments. Okay, the iron can be recycled. In other words, reused, or it can be lost due to trauma. Bleeding. Okay, so, and that's why women on a monthly basis, because of the menstruation, they lose a lot of the iron and then they become anemic because you need iron to produce heme groups, to produce hemoglobin, to produce red blood cells. Okay, and that's why women in particular have to make sure they have iron in their diet because we do lose it in through menstruation. You can also lose it through trauma, like if you have heavy bleeding, like a cut, or an accident that causes you to bleed, you can also become anemic. Okay. Now the pigment, this is really interesting because have we gotten to the digest? Yeah, we have. So the pigment goes on to the liver, okay, and it goes through the process of it goes to the liver. And then finally, it's going to be excreted as fecal matter. Fecal matter. And that's why there's kind of like a color to fecal matter. Okay. So it goes through an interesting process of how the pigment, when it goes to your liver, it becomes bilirubin. So it starts off as biliverdin, gets converted to bilirubin, and then exits as bile. Okay. So pigments, then the pigment goes to the liver. So the pigment that leaves to go to the liver is called biliverdin. I don't know that I require you to know this level of detail, but just in case you're curious. Biliverdin, and then once it goes to the liver, it gets converted to bilirubin, and then when it exits as bile. But in digestive system, you learn about bile, right? So you might as well know where it came from. So the bile came from this conversion from biliverdin as the pigment in the heme group. Okay. And that's why, so when it goes to the liver, newborns where the liver is not quite functioning yet, that pigment tends to stain their eyes and their skin, and they have a condition called jaundice because they're not able to process the bilirubin into bile. Okay, so it's really interesting how some small events here can cause diseases in us. And it's a very simple process. So the pigment from the red blood cells leaves as biliverdin, goes to the liver, gets converted to bilirubin. The bilirubin gets converted to bile, and bile is what is excreted into the digestive system and then is excreted as fecal matter. Okay, so that's pretty much the life cycle of a red blood cell. Any questions? It's kind of cool. Okay. Then the next thing, letter B, then A was erythrocytes, B is going to be plasma. For plasma, you want to make sure you know the function. I'm going to move the chat screen. You can't make it any smaller. Okay. So in plasma, you want to make sure you know the function of plasma, and that's where it transports gases, transports nutrients, transports waste products, and so on. You want to make sure you know the components of plasma, and you know that it's mostly water, and then it also has plasma proteins. Then there's tons of plasma proteins. If you look at your textbook, it has a long list of plasma proteins. We're just requiring you know the three basic ones. Okay, so there's three basic plasma proteins that I want you to know. What are they? Let's start with one. Name a plasma protein for me. Yeah, perfect. Fibrinogen. And I love that you use the term fibrinogen too. So fibrinogen, and this is the plasma protein that you would find in plasma. And notice that ogen right here. Whenever you see the term ogen, it means inactive. Okay. So fibrinogen is the inactive form of fibrin. Makes sense because you don't want fibrin unless you need it. Okay, so fibrin is a plasma protein that has clotting action. It can cause clots. And so you don't want clotting to occur in your body unless you need it. Because if it clots in your bodies, it could lead to like closure of your blood vessels, and that would be no good, right? Like strokes, heart attack, venous thrombosis. So clotting under normal conditions is not good. And that's why we have the inactive version of it in our bodies. And when there's trauma to that area, like if there's a cut in that blood vessel, then there are proteins on the outside that go in and activate the fibrinogen into fibrin, and that's what causes clotting, and hopefully it seals up that area that you have a cut along your blood vessel. Okay, but that's fibrinogen. What's another plasma protein? Because you have to know 3 of them. What's another one? Because you have to know 3 of them. What's another one? You're welcome. Thank you, albumin. So my dad used to go for paracentesis all the time. His liver wasn't quite working, so he was building up fluid in his abdomen. Okay. And after they took out all that fluid, they would have to give him a replacement of albumin. Okay. Because once they remove all that fluid from his body, his blood vessels couldn't manage the movement of water in and out of the blood vessel. So they had to give him albumin. Okay, so that's what albumin is. Albumin is a plasma protein that controls or regulates osmolarity. In other words, the movement of water, and viscosity, how thick blood is. Okay, what's a third plasma protein? Third one, last one. I mean, not the last one, because there's a lot of them, but last one of the three that we're making you know for this class. Yeah, okay. So globulins, okay? So globulins, there are three main types, okay? So under globulins, there are three main types. There's alpha and beta. And I lump them together because alpha and beta globulins are similar in that they're transport proteins. They transport things in plasma. But you also have a third type of globulin. And I want you to notice this because in the script, they didn't really elaborate on this, but it's on it's on your quizzes and exams. Okay, so I want to elaborate further on this. So in the globulins, the alpha and beta transports elements in the plasma, but you also have gamma globulins. And the gamma globulins are, think of them as your antibodies. Okay, and antibodies, what they do is that they're proteins that identify non-self. In other words, things that aren't you. So you can think of this as your immunity. Okay, so those are the three plasma proteins that you would find in blood. Okay, or in plasma to be specific. Then the third main component of blood is leukocytes, white blood cells. There's a mnemonic to kind of help you remember the components of white blood cells, and that's never let monkeys eat bananas. So never let monkeys eat bananas. It's nice because it gives you the first letter of the white blood cells. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Okay. So because I'm running short on time, I'm gonna have you, because this is easy one to look up. I'm gonna have you look up and tell me what they are and what do they fight? What's their main purpose? And I'll start with one for you. So neutrophils, what they do is that they undergo a process called phagocytosis. They eat things that shouldn't be in your body. So they're phagocytes that undergo phagocytosis. So they eat things, okay? And specifically, they target bacteria. So if you have bacteria in your body, you will start to see an increased level of neutrophils. Remember, leukocytes are supposed to be less than 1%. So if I see a lot of neutrophils in the body, that means that that person has some type of bacterial infection going on. Okay, so I want you to do the same for lymphocytes, monocytes, eosinophils, and basophils. Okay, another mnemonic to use is Benji, because the white blood cells are broken down into the types. Okay, we have granulocytes and we have agranulocytes. Anytime you see the word A in front of something, it usually means without. So these are the white blood cells that have granules inside of them, the granulocytes, where the agranulocytes are white blood cells that don't have the granules inside of them. And a good way to remember that is Benji. Basophils, eosinophils, neutrophils are granulocytes. So they have, so here's a neutrophil. Neutrophil has a, multilobular nucleus. And then they will have these granules inside of them. And that's what makes them granulocytes. Where monocytes down here have this pretty decent sized nucleus, but there's no granules inside of them. So that's a distinction between granulocytes and agranulocytes. Okay. Then let's take a look at blood flow. Oh, now we're going to move on to the heart. Oops. So the heart, make sure you know the structure of the heart and the function of the heart. And when I study these, I kind of study blood flow with it because they go hand in hand. Okay. So one really good suggestion is just to draw it out. Let's draw it out with, what color should I make my heart? Let's make it, I feel like, let's do green. I want a green heart today. Just draw and label. That's for me, that was the best way to learn anatomy and physiology. Green. Okay. So when you're going through this, let's do the chambers first. What are the two main chambers of the heart? There is a right and left. What are the two main chambers first? Yep. So you have the atriums and you have the ventricles. So, excuse me, water. I have the atriums, which are the ones on the top, and their function is to receive blood. okay? And they're going to receive the blood. So A1 is going to be the right or left? What's A1? And see if you remember your anatomical position. Yes, very good. That's going to be the right atrium. A2 becomes the left atrium. And B are the ventricles. And the function of the ventricles are to pump the blood out of the heart. So it makes sense that which one's more muscular, atriums or ventricles? Which one's more muscular? The ventricles, right? And that's why all of this space right here, this is all muscle. This is all muscle. Because the ventricles have to pump the blood out of the heart, okay? So I have B1 and B2, and it's the same thing, right? B1 is going to be the right ventricle, and B2 is going to be the left ventricle, okay? The next one that we're going to do are the blood vessels. And at this point, we're going to do it in order of how blood flows through the heart, okay? So first, we have this. We have 1A and 1B. 1A and 1B. So 1 is what? What's 1? The heart receives deoxygenated blood from the body. Yes, from the vena cavas. And 1A is the superior. And 1B is the inferior vena cava. Then it dumps the blood. Yes, it can. Oh, that's too thick. A little bit less thick. Make it a little bit transparent. So it goes through. Deoxygenated blood goes into from the vena cava to the right atrium. And then from the right atrium, it's going to pass by this valve right here. And we'll get to that valve in a little bit. Enters into the right ventricle. From the right ventricle, it's going to pass through this valve. And then it gets into this blood vessel. And we're going to call that blood vessel number 2. And what is number 2? What's number 2? That's correct. The pulmonary trunk. Anytime it's associated with the lungs, it's usually called pulmonary. And this is the pulmonary trunk. And don't forget the trunk, because anytime you have a trunk, it's going to split. And you can see that it splits right here. As a matter of fact, it splits into this part, 3A and 3B. And number 3 is what? It splits into the pulmonary what? Because it's leaving the heart, it's going to be the pulmonary what? Yes. Arteries always leave the heart. So this is going to be the pulmonary arteries, okay? And 3-1 is going to be the, 3A is going to be the right pulmonary artery. And 3B is going to be the left pulmonary artery. And then from there, it's going to go to the lungs, pick up oxygen, drop off the carbon dioxide, and the carbon dioxide you breathe out. So this is connected to the respiratory system. Then oxygenated blood goes back into the heart through four. I try to stick with the colors. So 4A, and this is going to be 4B. So what's number 4? Now it's going back to the heart from the lungs. So if it's coming from the lungs, you know it has to have pulmonary. But now it's going towards, yes, thank you, pulmonary veins. And the pulmonary vein has 4A and 4B. So 4A in this instance is going to be what? Yep, Irene said it. It's going to be the left pulmonary vein. 4B is going to be the right pulmonary vein. And let's show that here with the flow. So, oh, let me finish with this. Goes out to the lungs, is going to come back from the lungs through the pulmonary veins, the right and left pulmonary veins, into the left atrium. From the left atrium is going to pass by this valve into the left ventricle. From the left ventricle, it's going to move up this valve and into this area right here. This whole area is called, what number am I on? 5A, 5B, and 5C. What's 5? What's 5? Going to be the aorta. 5A is going to be the ascending aorta. 5B is going to be the aortic arch. 5C is going to be the descending aorta. Now 6, 7, and 8 is next. 6, so branching off of the aortic arch, I have 3 branches. 6 is going to be the brachiocephalic trunk. And it's called the brachiocephalic trunk because you would expect that it branches further, and it does, into 6A and 6B. So 6A off of the brachiocephalic trunk is going to be the right common carotid artery. Oh, sorry, that's the subclavian. That's 6B that I'm doing. 6A is going to be the right subclavian artery. 6B is going to be the right common carotid artery. Okay. That makes 7 then. The left common carotid artery. And 8 would be what? What's 8? If they're kind of mirror images of each other. What's 8? So let's try that again over here a little bit. That it doesn't, it's easier to see. So that would be 5B. And this would be 6, 6A, 6B, 7, and 8. So you can see that they're kind of mirror images of each other. So if 6B is the right common carotid going up through the neck on that side, 7 is going to be the other side. So the 7 is the left common carotid going up to the neck. Carotid means neck. If 6A is going to the right arm, 8 goes to what? Yes, that's going to be the left subclavian because it's going to go to the left arm. Okay, and those are the blood vessels. So then the last thing we want to cover are the valves. Oh, I'm out of labeling. Let's go back to, no, yes, let's go back to letters because we've only done A and B. So here's C, here's D, C1, C2, D1, and D2. Okay, so you have two main types of valves. You have the atrioventricular valves, atrioventricular valves, and just like their names, they're between the atrium and the ventricle. And that's going to be your C1 is going to be the right atrioventricular valve. I'm just going to use abbreviation for short because I'm running out of room. So right AV valve. Now the right AV valve, if you look right here, it has 3 flaps. You see the 3 flaps right here? 1, 2, 3. So it's also known as the tricuspid valve. So the right AV valve is also known as the tricuspid valve. Cusp meaning flaps, tri meaning 3, because it has three parts. Okay. C2 is going to be the left. C2 is right here. C2 is going to be the left AV valve. And it's also known as what? Because how many flaps does it have? 1, 2. So you guess its name. What do you think it's also known as? Yeah, the bicuspid valve. And if you talk to really old doctors, it used to be called the mitral valve. So C2 has 3 names. C2 can be known as the left AV valve or the bicuspid valve or the mitral valve. I will take any of those 3 on the exam. Okay, but you have to do it correctly. You can't say left bicuspid mitral valve. You know, you have to use the names correctly. It's either 1, the left AV valve, or 2, the bicuspid valve, or 3, the mitral valve. Okay, so that's the first type of valve. The second type of valve is D. Now D is called the semilunar valves. The semilunar valves look like half moons. So they look like this. They don't have flaps. They just have these half moons. And that's why they're called the semilunar valves. Okay, D1, D2. So D1, because it's going to the lungs, is called the pulmonary valve. It used to be called the pulmonary semilunar valve because that was so long, they just shortened it to the pulmonary valve. D2 is going to the aorta, so it's called the aortic valve. Same thing, it used to be called the aortic semilunar valve, but people just shorten it to the aortic valve. Okay, and that's it for the heart and the blood flow. So it's really, it's less complicated if you do you overlap the parts as well as the blood flow, because it makes a lot of sense once you do that. I'm going to move my chat screen over to this side a little bit. So this afternoon, we talked about the cardiovascular system, which include the blood, heart, and blood vessels. We went through kind of the functions, components, red blood cells, and then plasma, and then the heart. And that's pretty much where we left off. Okay. So we didn't get a chance to go into much detail about the heart besides the structure and blood flow, But do make sure you do the cardiac conduction and EKG. A lot of you did really great videos on that, so make sure you watch each other's videos. Now for blood vessels, I noticed one thing about the blood vessels that with the video, to those of you who did the video on the blood vessels tended to stay more of the general when you really do need to know more of the specifics, okay? So make sure you know not you know the three main types of blood vessel, but also the layers and then how the structure of those layers contribute to the function and characteristics of those blood vessels. A good way to learn that is to make a cognitive map. So I actually made a cognitive map for the class here. And what I would recommend you do is that you keep this as a key, but try to do this on your own. So here you can see that I have the functions of each type of blood vessel as well as the layers. Now the layers between the arteries and the veins, they have the same 3 layers, but within those 3 layers, there are slight differences. And those differences contribute to the unique characteristics. Okay, so make sure you can kind of tease out the differences for those. And then there's also the capillaries below. Let's see if I can scroll down. There we go. See a little bit of that. Oh, okay. So that's the cognitive map for the blood vessel. So you want to make sure you understand that as well. Okay. All right. So for today or for this evening session, we're going to focus on the respiratory and the digestive. I think students have usually a lot of requests about the ventilation and gas exchange, and that's what I'll be focusing on. And then for digestive, the chemicals usually is what gets students, so I'll be focusing on that. All right, so with the respiratory system, make sure you know the functions of the respiratory system as well as the structure. Okay, and that's where I had you do the worksheet for this week right here for the respiratory system. So just make sure you review your worksheet. I do cover the worksheet and in my lecture. So what I did is that what happened is that during COVID, my in-class version was made to go online, and I recorded those sessions and then I put them into the announcements on Canvas. So if you want to watch those kind of in class but online lectures, make sure you take a look at that. So there's the lecture videos, and these are supplemental. So these are not the ones that are on your your main Canvas page. So these are the supplemental lecture videos. Let me move the chat. Has any one of you had a chance to take a look at those? Until announcements. Great. Okay. So in there, I spent a lot of time talking about the sensory organs as well as the tissue linings. Now, the tissue linings in previous classes, students struggle with that because they try to memorize it. And here's the problem. It's a lot of detail to memorize. Rather than memorizing it, you should try to understanding it. So because when you try to understand it, it helps to remember what it is. So for example, from the upper respiratory tract, the majority of the upper respiratory tract is lined with pseudostratified ciliated columnar, epithelial columnar. Okay, and so that kind of tells you that it because of it has mucus cells in there as well as goblets, the goblet cells which produce mucus, and the cilia, which then helps to move things along. So that's an important function of the upper respiratory tract, that you have the mucus and the cilia that helps to move things along. Then as it transitions into your lower respiratory tract, it changes from the pseudostratified columnar epithelium to more of the simple columnar, to then the simple cuboidal, and then to the squamous, which allows for the exchange to happen because it's a thin cell. Okay, so when you look at the tissue lining, okay, Try to connect the lining with function. And then at that point, it would be more of understanding it, and it just makes sense rather than memorizing the transitions and changes of the tissue lining. Okay, then ventilation and gas exchange are the two main concepts that we cover in the respiratory system. Okay. And those are the two that I'll spend some time on. So we'll start with ventilation. And ventilation is essentially how we breathe. And breathing includes inspiration, breathing in, and expiration, breathing out. Okay, so before we go through the ventilation, there are three main concepts that you have to understand to be able to move forward with ventilation. One of the concepts is Boyle's law. Can someone tell me what Boyle's law is? It's it's really complicated, but we kind of only require you know the basics of it. So what's the basics of Boyle's law? What does Boyle's law say? Okay, so Boyle's law, just as a reminder, tells you about the relationship between volume and pressure. You can think of pressure as the force. Okay, so it says that when one goes up, the other one goes down, so that the relationship between volume and pressure are inversely proportional. Inversely proportional. Okay, so if volume goes up, what happens to pressure? Yes, exactly. So space is related to pressure. Thank you, Sean. Okay, and likewise, when volume goes down, pressure goes up. Okay, a really nice analogy of that is if I had a can of soda. Okay, you can see that with the can of soda, if you shake it up, when you cause pressure, when you cause force, the molecules inside the soda bump against each other and the pressure increases. Okay, so pressure is high because volume is low. There's not a lot of space. But if I took that same amount of soda and I put it into a gallon of milk, now what I've done is I've increased volume. So what happens to the pressure? The pressure is going to decrease because now the molecules in here have more space to travel around, so it's less likely to bump into each other and create pressure. Okay, so that's what Boyle's law say, is that volume and pressure are inversely proportional. And it makes sense. So if I put the same amount of liquid into a can of soda versus into a gallon, it's going to have different pressure. Okay. All right. Then the second concept is diffusion. And this one, all of you should know because you learned about it in the chapter on cells. What does diffusion tell us? That things move from what to what? Perfect. It's a movement from high to low concentrations. Okay. Then the third thing that we have to know about ventilation, besides the Boyle's law and diffusion are the main muscles that play a role in ventilation. The main muscles, what are the main muscles that play a role in ventilation? There's several, but we kind of focus on two. What are the main muscles that play a role? They are they are skeletal muscles. Yep. But what particular muscle are we talking about? Yeah, exactly. So we're talking about the diaphragm. Thank you for both of you. The diaphragm and the intercostal muscles. Okay, so these are the two main muscles in breathing, the diaphragm and the intercostal muscles. The intercostal muscles are the ones in between your ribs, hence intercostal. Intercostal muscles, and then your diaphragm. Okay. All right. So knowing this, you can draw this. Okay. So here's your thoracic cavity. Now your diaphragm is naturally curved like this. Okay. So what's going to happen? A lot of students think that you breathe in and that causes your thoracic cavity to expand, but that's not the truth. Actually what you have to do is that you have to change, the muscles have to react a certain way for the air to move in or out. Okay, so a lot of students, a misconception is that the air moves in or out, and then that causes a change in your lungs. Okay, but that is not true. It's actually the reverse. So for inspiration to occur, first the space has to change. Okay, so the thoracic cavity has to change. And then that's what drives the air to move out or in. Okay, but if we were looking at inspiration, what does the diaphragm have to do? Does it have to contract or does it have to relax for inspiration to occur? So if it relaxes, it's going to move downwards like this. Okay, so that's the diaphragm relaxing. Oh, I'm sorry. I drew it wrong. That's when it gets shorter. So when the diaphragm relaxes, it's actually going to get longer. Because remember, when something relaxes, it gets longer, right? So when the diaphragm relaxes, it's going to get longer. Because you can see the difference from its normal state right here. And if you were to measure the distance, it's going to be shorter than this top one right here. So that's showing you your diaphragm relaxing. So if your diaphragm relaxes, the volume decreases because now the space is, oops, let's do, now I want to make it thicker. Let's do it here. So here's the space. So volume decreases, which causes pressure to increase. Now, if you compare that to atmospheric pressure, okay, so the atmospheric pressure is about how much? What's the atmospheric pressure? So what's the atmospheric pressure? Nope. Oh, sorry. Let me get that correctly. So, yep. So there you go. Okay. So the atmospheric pressure is about 760, okay, millimercury. Millimercury. Okay. So when the volume decreases and the pressure increases, the pressure goes about 763. Okay. So when the pressure increases to 763, at this point, the pressure in the lungs is higher than the pressure in the atmosphere. Okay. And we know from diffusion that it will go from where it's high to where it's low. So the air, when your diaphragm relaxes, is going to go in which way, in or out? Exactly. Okay. So when the diaphragm relaxes, what is the result of that? Expiration or inspiration? Mm, there you go, Savannah. Expiration. Because the air is moving out, right? So when you expirate, you're exhaling. So the air moves out through expiration. Does that make sense, Sean? And it's okay if it doesn't. Oh, okay, good, good. Okay. So when the diaphragm relaxes, it pushes up a little bit because it gets longer, right? And so when the diaphragm relaxes, that always leads in a process called expiration when you're exhaling. Okay. Now, if the diaphragm contracts, then it gets shorter. And do you remember when I said that the diaphragm is normally curved upwards like this? Okay. So when the diaphragm contracts, it gets shorter. And when it gets shorter, it pulls downwards. Okay. So when it relaxes, it pushes up. When it contracts, it pushes down. So when it pushes down, when it gets shorter, because you can see like the distance between here to here with a straight line, it's shorter, right? And that's what happens when muscles contract. So when muscles contract, the diaphragm straightens out, gets shorter. And when it does that, now the volume changes, and the volume is going to include this whole area. Okay. So the volume now increased. And if the volume increases, that causes what to happen to the pressure inside the lungs? Yeah, it decreases. As a matter of fact, it goes down to about 756 millimercury. That's two M's in there. Okay. So the diaphragm contracts, the pressure inside the lungs goes down to about 756 millimercury. When you compare the pressure of the lungs to the pressure of the atmosphere, now which one is higher? The lungs is not higher, actually. So the pressure of the lung, there we go. Sorry, I forgot to put the L in there. So the pressure of the lung is now 756, where the pressure of the atmosphere is 760. So the pressure of the atmosphere is higher, right? And we know that in diffusion, things go from high to low. So in the case that the diaphragm contracts, okay, the air is going to go from where it's high to low. So in other words, from the outside into the lungs, okay? So when the diaphragm contracts, that's going to result in inspiration. Okay. So that's why you have to have a change in the muscles first, and then that causes a change in the volume and pressure. And then it follows diffusion to move the to move the air in or out of the lungs. Okay. Questions? So true or false? In order for inspiration to occur, the pressure inside of the lungs has to be equal to the pressure in the atmosphere. True or false? Perfect. Perfect. What would make that statement correct? The pressure inside of the lungs must be what to the pressure outside in order for inspiration to occur. Yes. Wait, less than. There you go. Okay. So in order for it, yes, that's right, Anna. So in order for inspiration to occur, the pressure in the lung must be less than the pressure in the atmosphere. Yep, Sean, you got it. Absolutely. Okay. So it takes some thinking through. Don't like, even I had to stop and think about it, because it's so similar between expiration and inspiration. So I really have to stop, think it through, and then answer the question. Okay. But if you go through this process, you will always get it right. Okay. So first start with the muscles, then with the pressure, volume and pressure, and then follow the diffusion, and that will always lead you to the right answer. Okay. So when the diaphragm contracts, volume increases, pressure decreases, and then when I compare the pressure of the atmosphere to the pressure of the lungs, then I know that the pressure in the lungs is lower when the diaphragm contracts, is lower than that of the atmosphere, so air moves in, and thus it leads to inspiration. Okay. Questions? That was kind of fun. Okay. It's like a logic mod, you know? All right. Moving on. So now we have gas exchange. So in gas exchange, there are two parts of gas exchange that I'm requiring you to know. One is pulmonary gas exchange, and the other one is systemic gas exchange. Okay, so one is pulmonary. So in other words, the lungs, and the systemic is the body. Okay, so let's go through gas exchange. So if we're looking at the lungs, where does gas exchange happen in the lungs? Perfect. Thank you. Okay, so I'm going to zoom in at the alveoli. Here's the alveoli. Okay. So we mentioned that when you inhale, air moves in, and that air, what's the composition of air? Air is actually mostly what? You had to do this on your worksheet. Yes, perfect. So air is mostly nitrogen. Only about 20% is oxygen, but it's more oxygenated than, of course, what deoxygenated blood would have. So even though I'm drawing oxygen here, you should know that air is actually mostly nitrogen. But comparatively, or what we use partial pressure to define, oxygen is higher in the alveoli when you first inspirate. Okay. Now, coming from the heart, what blood vessel takes blood to the lungs? What blood vessel? The pulmonary, nope. The pulmonary vein will go back to the lungs. There you go. So this is going to be the pulmonary arteries. Perfect. So the pulmonary artery is going to be high in what? CO2 and low in O2. Okay, so the pulmonary arteries, which carries blood away from the heart to the lungs, is going to be high in carbon dioxide and low in oxygen. Okay, so the pulmonary arteries eventually is going to become capillaries. Okay, and capillaries allow for gas exchange to occur between the capillaries and the alveoli. So in order to understand this, we have to take a look at this membrane right here. Okay, so let's take a look at the respiratory membrane. So the respiratory membrane consists of blue green, the alveoli, and the alveoli has what cells that allow for exchange? The alveoli has what type of cells that allow for the exchange? So if you review, the alveoli has three types of cells. The alveoli has great squamosal cells, and the great squamosal cells produce surfactant. It has the dust cells, and the dust cells are macrophages. And then it has the simple what? So kind of review the three cells of the alveoli in your head. Yeah, it is an epithelium, but what kind of epithelium? What shape makes sense for a diffusion to happen? Should it be cube shaped, thin and flat, or tall and thin? Yes, thank you. It's got simple squamous cells, and that's what allows for diffusion between the alveoli. Now you have the blood vessel, and you know it's a capillary, right? So the capillary is going to have what type of cell? It's also simple. Kind of review what you presented on in in your video on the blood vessels. Those of you that did the compare and contrast. Capillaries have a layer of what? Simple what? also simple squamous. Okay. And then there's a basement membrane between the two. And that's what makes up the respiratory membrane. So you've got one, the alveoli, followed by two, the basement membrane, followed by three, the capillary, simple squamous of the capillary. Okay. So in the blood vessel, in the capillaries, or transitioning from pulmonary arteries to capillaries, you're going to have lots of CO2. Where in the alveoli, you're going to have O2. Okay, so you're going to have an exchange happening. The CO2 moves into the alveoli, and the oxygen moves from the alveoli into the capillary. So what ends up exiting as the pulmonary vein, pulmonary vein, now because of the exchange, it is high in O2 and low in CO2, and this is what goes back to the heart. Okay, so that's pulmonary gas exchange. Pulmonary gas exchange happens between the respiratory membrane, which consists of one, the simple squamous of the alveoli, the basement membrane, and the simple squamous of the capillary. Does that make sense? Questions? Take a second to look at that. Let me know if you have any questions. So that's the pulmonary. Let's do the same thing again for the systemic. Actually, I only have 20 minutes left. I wonder if I want to go into. Would you prefer for me to do the systemic, or would you prefer for me to go through the digestive? With the systemic, you're just reversing it, and now between the blood vessel and the cells of the body instead of the alveoli. Digestive, okay, right. So you would kind of copy what I drew here, but instead of the alveoli, you would put the cells of the tissue in the body, and then you just reverse everything, okay? But I want to make sure I spend some time on the digestive as well. All right, so for the digestive, you want to make sure that you know the function of the digestive system. Let's just quickly list off some functions. What are the functions of the digestive system? My favorite part. It all starts out with what? Yes, ingestion. Ingestion followed by digestion. And then you have absorption. And then compaction and defecation. Look at that. You even wrote it in the correct order. Okay. So you have ingestion, digestion, absorption, compaction, and defecation. Great. All right. So you also want to make sure that you can describe the digestive tract. Do the same thing that we did for the respiratory tract, following how it goes from the nose to the lungs. Here, you'd go from the mouth to the anus. So if you think about it this way, the digestive tract goes through the body. It's one continuous tube. Okay. And as the food moves through the digestive tract, is it technically in your body or is it moving through your body? So the alimentary canal allows for things to move through your body, but not technically be in your body. Okay, exactly. So things that are in your stomach, in your intestine, they're technically not in your body. And that's why we have to have a process called absorption. So until it gets absorbed and actually moves into the bloodstream of the body, it technically is just moving through the body, but not into the body. Okay? All right. So in addition to knowing the alimentary canal as it transitions from the mouth to the anus, you also want to know about the layers. Okay. And on the test, I can ask you to go from superficial to deep or deep to superficial. Okay. So you want to make sure you read the question carefully. I'm sorry, I know green is really hard to say. I should have used blue. Okay. So make sure you read the question carefully as to whether I'm asking you to go from deep to superficial or from superficial to deep. Because that's going to make a difference between whether you're going from mucus, mucosa, submucosa, to what? Muscularis, to serosa, or the reverse. Okay? Then go through, starting from the mouth. Now, the mouth is complicated because it's got so much accessory organs. It's got the tongue, the lingual papilla, the lingual frenulum, palate, uvula. Know all of these structures and the function. Know the teeth in detail. So, for example, what's the root versus what's the neck versus what's the body of the tooth? Know the glands, where are they located and what do they secrete? Like the sublingual gland, the parotid gland, and the submandibular glands. Okay. Follow the movement of food through the pharynx, into the esophagus, to the stomach, to the stomach. Where's my intestine? Now to the small intestine, to the large intestine, and then also know the accessory organs. Okay. And a good way to learn these is to do the cognitive maps, to do that list that you did for the respiratory, like 1, 2, 3. And then as you're going through that, what are they? What layers do they have and what characteristics do they have? Okay. So with the remaining time that I have left, I'd like to focus that on the chemical digestion, if I may. Okay. So here you can see that I started a list in terms of if you were to eat an almond joy, and I chose almond joy because it's my favorite candy, and two, it has all the important components. So the almond in an almond joy is the protein. The coconut is the lipids, and the chocolate represents the carbohydrates. Okay. And that's why I chose an almond joy, because it kind of has all the nutritional needs. And that's why it's my favorite. Okay. So at the mouth, due to the tongue, the teeth, and the salivary glands, this is where ingestion happens. And that's followed by digestion. Now, in the mouth, two forms of digestion happen. The teeth and the tongue allow for physical digestion or mechanical digestion in a process called mastication, meaning chewing, and that's why you have the masseter muscle in your mouth that helps you to chew. The teeth grinds the food apart, and that's part of the mechanical or the physical digestion. But in the mouth, you also have chemical digestion because the salivary glands produce saliva. And in saliva specifically, you have salivary amylase. Okay? If you haven't noticed it by now, anything that ends in an ACE means it's an enzyme. Okay? So salivary amylase is an enzyme that breaks down amylase, which is carbohydrates. Okay? So salivary amylase is the chemical, the enzyme that breaks down carbohydrates. So the first thing that's chemically broken down is going to be the carbohydrates. And you notice that. You should notice that, right? So if you eat a meal with rice versus with meat, when you eat the meal that's heavily on with rice or carbohydrates, you get hungry faster. And the reason for that is because you break it down earlier on. You break it down right when it goes into your mouth and encounters the physical breakdown as well as the salivary amylase. You can physically break down the meat, but it's not going to be chemically broken down until it gets to your stomach. And that's why when you eat meat, you feel fuller longer. Where if you eat carbohydrates, you don't feel full as long because already it's starting to be broken down in the mouth. Okay, questions? All right. Then it's going to continue along the alimentary canal from the mouth to the oral pharynx and the laryngeal pharynx. Okay. From the laryngeal pharynx, it's going to go to the esophagus. And all these are just pathways. There's not digestion happening. So it's just a pathway until it gets to the stomach. Okay. So the stomach looks something like this. The opening of the stomach is the cardiac. The dome of the stomach, this portion right here, is the fundus. Then the middle portion right here is the body. And the last portion is the pyloric. So P, cardiac, fundus, and body. So those are the parts of the stomach. Okay. Now, the stomach is going to allow for the physical breakdown, to the churning that happens to the contractions of the stomach, but mostly we focus on the chemical breakdown. Okay. In the stomach, it produces a substance called pepsinogen. You will want to know what produces pepsinogen. So the stomach has several cells. What are the cells that you find in the stomach? Like the chief cell, okay, like the parietal cells, and so on. So you will want to know what cell produces what, okay? But if we're looking at just chemical, you've got the pepsinogen. And like I said this morning, anything that ends in ogen means what again? Who remembers from this morning? If it's a term that ends in ogen, like fibrinogen, what does the ogen mean? It means it's inactive. Okay, so if it ends in ogen, it means it's inactive. Like fibrinogen is the inactive form of fibrin. Okay, pepsinogen is the inactive form of pepsin. Okay, so pepsinogen is going to be converted to pepsin when hydrochloric acid is present. So in the presence of hydrochloric acid, the pepsinogen is going to be converted to pepsin, and as a chemical, pepsin breaks down proteins. Okay, so pepsin is going to break down the protein. In other words, it's going to break down the almond. It's going to break the proteins down into peptides. It's going to break down the protein, which is a large structure, into the polypeptides. Okay. The hydrochloric acid helps to convert the pepsinogen into pepsin, but the hydrochloric acid, because of the acidity, its function is more to kind of clean the bacteria that's in the food. So it helps to break down the bacteria in food. Okay. Let me move my slide down a little bit. You also have gastric lipase and to some point salivary lipase. So the salivary lipase is actually a component of saliva, but it doesn't activate until it has a certain pH requirement, which it meets in the stomach. So the salivary lipase is produced by the salivary glands, but is not activated until it gets into the stomach. So both the gastric lipase and the salivary lipase is going to begin partial breakdown of lipids. Okay. From there, the food is going to go to the small intestine. So the small intestine has the following regions: duodenum, jejunum, and ileum. I have a mnemonic that helps me remember that, and that's don't jump in. Don't jump in. Duodenum, jejunum, and ileum. Now, different things happen in different areas, okay? If we're looking at digestion, the majority of digestion in the small intestine is going to happen along the duodenum. By the time the food moves through the jejunum and ileum, there shouldn't be that much more digestion happening, okay? So digestion primarily happens in the duodenum. And the reason for that is because after the stomach, it's going to pass through several accessory glands that secrete. So at this point right here, between the stomach and the small intestine, you have the accessory glands that play a role here. And the accessory glands include the liver, which contributes the bile, the pancreas, which contributes a ton of chemicals. Okay, so you may want to make a list of all of that. Okay. Oh, and I wrote it right here. Okay. So some examples of the chemicals that is produced, okay, by the sensory organs and secreted into the small intestine along with the food that has been broken down already includes pancreatic amylase. What do you think pancreatic amylase breaks down? Yep. You've got pancreatic lipase, as well as the bile. Both of these target what? Fats. Perfect. Then you have the chymotrypsinogen, which technically is inactive, but will convert to chymotrypsin. Then you have the procarboxypeptidase, which converts to the carboxypeptidase. And then the trypsinogen, I'll move down the side anymore. Nope. Trypsinogen. Oh, sorry, the peptidase. No, I already did the peptide. Trypsinogen, which converts to trypsin. And all of these target what? Yeah, protein. So at this point, because we already had the pepsin break down to the polypeptides, at this point, the polypeptides are going to be broken down into what? Yep, you can have dipeptides, but eventually it has to become what? Yeah, amino acids. Okay, all right. So that's it for our review today. Do you have